Right now, as you listen to these words, billions of neurons in your brain are firing in precise coordination, processing language, forming memories, maintaining your awareness of the world around you. But in a few hours, something extraordinary will happen. Your consciousness will fade, your body will become still, and your brain will embark on one of its most vital and mysterious journeys. Sleep occupies roughly 1/3 of human existence, yet it remains among the most enigmatic processes our bodies perform. Every night, without conscious effort or deliberate instruction, your mind orchestrates an intricate symphony of electrical activity, chemical cascades, and cellular maintenance that rivals the complexity of any waking thought. While your body lies motionless and your eyes remain closed, your brain enters distinct phases of heightened activity, each serving specific biological functions that simply cannot occur during consciousness. This nightly disappearance into unconsciousness is not mere rest, but rather an active, purposeful state essential to memory, health, and survival itself. From the consolidation of today's experiences to the clearance of metabolic waste, from hormonal regulation to the vivid theater of dreams, sleep represents a masterpiece of neural coordination. The more we discover about these hours of apparent inactivity, the more we realize they may contain the brain's most sophisticated performance of all. Where are you listening from right now? And what time is it where you are? It's truly amazing to have you here with me as we explore this fascinating subject together. Let's get into the video. The revelation that sleep was not simply an absence of consciousness came in 1929, when a German psychiatrist named Hans Berger made a discovery that would revolutionize our understanding of the sleeping mind. Using primitive electrodes placed on the human scalp, Berger detected rhythmic electrical oscillations emanating from the brain. These brainwaves, as they came to be known, continued even during sleep, but their patterns changed dramatically as the night progressed. What Berger had stumbled upon was the first glimpse into sleep's hidden architecture. Far from being a uniform state of unconsciousness, sleep revealed itself as a dynamic, cycling process with distinct stages, each characterized by unique patterns of brain activity. This discovery launched decades of research that would map the nightly journey of the human brain through different phases of neural activity. Modern sleep studies using electroencephalography, or EEG, have revealed that sleep consists of two fundamentally different states: non-rapid eye movement sleep, known as NREM, and rapid eye movement sleep, called REM. These states alternate throughout the night in predictable cycles, each serving distinct biological functions that are essential to brain health and cognitive performance. NREM sleep unfolds in 3 progressive stages, each representing a deeper level of unconsciousness. Stage 1 is the lightest phase, a brief transitional period lasting only 5 to 10 minutes, as you drift from wakefulness into sleep. During this stage, your brain waves shift from the alert beta rhythms of consciousness to slower alpha waves, and then to even slower theta waves. Your muscles begin to relax, your heart rate slows, and your breathing becomes more regular. You remain easily awakened during this phase, often denying you were asleep at all if roused. Stage 2 represents the onset of true sleep, accounting for roughly 40 to 50% of your total sleep time. Here, the brain produces distinctive patterns called sleep spindles and K-complexes. Sleep spindles are brief bursts of rhythmic brainwave activity, lasting half a second to 2 seconds, generated by the thalamus deep within the brain. These spindles appear to serve as sentries, blocking external sensory information from reaching the cortex and disrupting your sleep. K-complexes are large, slow waves that seem to suppress arousal and maintain sleep stability. During stage 2, your body temperature drops, your heart rate continues to slow, and you become much more difficult to awaken. Stage 3 represents the deepest phase of NREM sleep, characterized by slow, powerful delta waves that sweep across the brain like ocean swells. This deep sleep stage typically occurs during the first half of the night and is crucial for physical restoration and memory consolidation. During stage 3, your brain waves slow to just 1 to 4 cycles per second, compared to the 30 or more cycles per second seen during wakefulness. Your muscles are fully relaxed, your breathing is slow and rhythmic, and awakening you requires considerable effort. Blood flow to the brain decreases, while blood flow to muscles increases, facilitating tissue repair and growth hormone release. After cycling through these NREM stages, your brain enters an entirely different realm, REM sleep. This paradoxical state earned its name from the rapid, jerky movements of the eyes that occur beneath closed lids. Despite your body being virtually paralyzed by neural mechanisms that prevent you from acting out your dreams, your brain becomes almost as active as during wakefulness. Brain waves during REM sleep closely resemble those of an alert, awake brain, with fast, low-amplitude beta and gamma waves dominating the EEG recording. REM sleep typically begins about 90 minutes after sleep onset and occurs in increasingly longer episodes throughout the night. The first REM period may last only 10 minutes, while final REM periods can extend for 30 to 60 minutes. During REM sleep, your brain temperature rises, your blood pressure and heart rate increase, and your breathing becomes irregular and rapid. Most vivid dreams occur during this stage, though dreaming can happen during NREM sleep as well. This entire progression from stage 1 NREM through REM sleep constitutes one complete sleep cycle, lasting approximately 90 minutes in adults. A typical night contains 4 to 6 such cycles, though their composition changes dramatically as the night progresses. Early cycles contain more deep NREM sleep, while later cycles are dominated by REM sleep and lighter NREM stages. This shifting architecture means that most deep sleep occurs during the first third of the night, while most REM sleep happens during the final third. The master conductor orchestrating this nightly symphony is a tiny cluster of neurons in your hypothalamus, called the suprachiasmatic nucleus. This biological clock, containing only about 20,000 cells, receives direct input from specialized light-sensing cells in your retina and synchronizes your sleep-wake cycle with the 24-hour rotation of the Earth. The suprachiasmatic nucleus influences the release of melatonin from the pineal gland, typically beginning around 9 in the evening as darkness falls, signaling to your body that sleep time is approaching. Individual variations in sleep architecture are considerable and influenced by age, genetics, lifestyle, and health status. Newborns spend about 50% of their sleep time in REM, compared to only 20 to 25% in healthy adults. As we age, deep NREM sleep decreases significantly, and sleep becomes more fragmented with frequent awakenings. Some people are natural early risers, called larks, with earlier bedtimes and wake times, while others are night owls who prefer later schedules. These chronotype differences reflect genetic variations in circadian rhythm regulation. The discovery of sleep's complex architecture has profound implications for understanding how rest affects brain function. Each stage serves specific purposes. Light NREM sleep facilitates the transition to deeper states. Stage 2 sleep maintains unconsciousness and begins memory processing. Deep NREM sleep promotes physical restoration and memory consolidation, and REM sleep supports emotional regulation, creativity, and procedural learning. Disruption of any stage can impair these essential functions, highlighting why both sleep quantity and quality matter for optimal brain health. Understanding sleep architecture also reveals why timing matters. The brain's natural tendency toward deep sleep early in the night and REM sleep later means that shortened sleep typically reduces REM sleep disproportionately, potentially impacting emotional regulation and creative thinking. This intricate choreography of brain states represents one of the most remarkable aspects of human neurobiology, transforming our understanding of what it means to sleep. The brain speaks in electricity, and during sleep, this electrical language becomes a symphony of coordinated rhythms that would make any conductor envious. Each stage of sleep generates its own distinctive frequency patterns, brainwaves that can be measured and decoded to reveal the hidden conversations occurring between billions of neurons as they orchestrate the nightly maintenance of your mind. Delta waves represent the deepest voice in this neural choir, resonating at the slow, powerful frequency of 1 to 4 cycles per second. These waves emerge during stage 3 NREM sleep, sweeping across your cerebral cortex like massive ocean swells. Delta waves are generated primarily by the thalamus and cortex working in synchrony, creating the rhythmic, high-amplitude oscillations that define deep sleep. During this phase, your neurons fire together in remarkable coordination, with millions of cells alternating between active and silent states in unison. The presence of delta waves signals that your brain has entered its most restorative mode. These slow oscillations facilitate the transfer of information from temporary storage areas to more permanent memory locations, the process essential for learning consolidation. My research has shown that the strength and prevalence of delta waves correlates directly with memory performance. The following day, people who generate more robust delta activity during deep sleep demonstrate better recall of information learned before bedtime. Delta waves also coordinate the release of growth hormone from the pituitary gland, making deep sleep crucial for physical development in children and tissue repair in adults. The synchronized firing patterns that create these waves appear to trigger cellular processes throughout the body, from protein synthesis to immune system activation. Disruption of delta wave generation, whether through aging, sleep disorders, or medication, can impair both cognitive function and physical health. Theta rhythms operate at a faster frequency, typically 4 to 8 cycles per second, and play a crucial role in memory processing and emotional regulation. These waves are most prominent during REM sleep and the lighter stages of NREM sleep. Particularly during transitions between sleep stages. The hippocampus, your brain's primary memory center, generates distinctive theta rhythms that coordinate the replay of daily experiences during sleep. During theta-dominant periods, your hippocampus engages in intensive communication with the cortex, transferring memories from temporary storage to more permanent locations. This process involves the repeated activation of neural circuits that were engaged during learning, essentially rehearsing important information while you sleep. Theta rhythms also facilitate the integration of new memories with existing knowledge, helping to create meaningful connections and insights that may not have been apparent during waking hours. The emotional processing functions of theta waves are equally important. These rhythms help regulate activity in the amygdala and other limbic structures, potentially reducing the emotional intensity of stressful memories and promoting psychological well-being. Disrupted theta activity has been linked to depression, anxiety, and post-traumatic stress disorder, highlighting the importance of healthy sleep for mental health. Alpha waves, oscillating at 8 to 12 cycles per second, dominate during the transitional periods between wakefulness and sleep. These waves represent a state of relaxed alertness and play a crucial role in the sleep initiation process. As you begin to feel drowsy, alpha waves replace the faster beta waves of active consciousness, signaling that your brain is preparing for sleep. Alpha rhythms are generated primarily by the thalamus and serve as a bridge between conscious awareness and sleep. They help coordinate the gradual reduction in sensory processing that allows you to disengage from your environment. The presence of alpha waves during sleep-wake transitions also facilitates the synchronization of different brain regions, preparing neural networks for the coordinated activity patterns of sleep. Sleep spindles and K-complexes represent two of the most distinctive and protective mechanisms of sleeping brain activity. Sleep spindles are brief bursts of rhythmic brainwave activity, typically lasting half a second to 2 seconds and occurring at a frequency of 11 to 15 cycles per second, generated by the thalamic reticular nucleus. These spindles appear as sudden increases in brainwave amplitude that resemble spindles on an EEG recording. Sleep spindles function as neural gatekeepers, actively blocking external sensory information from reaching the cortex and potentially disrupting sleep. When sound, light, or other stimuli threaten to wake you, sleep spindles increase in frequency and intensity, working to maintain unconsciousness. People who generate more sleep spindles are better able to sleep through environmental disturbances and report higher sleep quality. K-complexes are large, slow waves that appear suddenly during stage 2 NREM sleep, resembling the letter K when viewed on an EEG recording. These waves represent brief periods of synchronized neural activity that help maintain sleep stability. K-complexes can be triggered by external stimuli or occur spontaneously, and they work in concert with sleep spindles to protect sleep from disruption. During REM sleep, the brain generates fast, low-amplitude gamma waves oscillating at 30 to 100 cycles per second. These high-frequency rhythms are associated with conscious awareness and complex cognitive processing during wakefulness, and their presence during REM sleep may help explain why dreams can feel so vivid and realistic. Gamma waves during REM sleep are thought to coordinate the integration of sensory, emotional, and memory information into the complex narratives we experience as dreams. The coordination between different brain regions during sleep represents one of the most remarkable aspects of neural function. The thalamus serves as the primary conductor of this sleep orchestra, using its connections to virtually every part of the cerebral cortex to synchronize brainwave activity across different regions. This tiny structure, no larger than a walnut, controls the flow of information between brain areas and determines when different regions should be active or silent. During NREM sleep, the thalamus helps generate the synchronized oscillations that characterize each sleep stage. It coordinates the timing of sleep spindles across different cortical areas and helps maintain the slow oscillations of deep sleep. The thalamus also gates sensory information, deciding which stimuli are important enough to potentially wake you and which should be filtered out to preserve sleep. The interplay between different brain regions during sleep creates dynamic networks of activity that shift throughout the night. During deep NREM sleep, the default mode network, associated with self-referential thinking and introspection, becomes highly active. During REM sleep, networks involved in visual processing, emotion and memory become strongly engaged, while areas responsible for logical reasoning and self-awareness become less active. This intricate choreography of brain waves and regional coordination demonstrates that sleep is far from a passive state. Instead, it represents a highly organized and purposeful process that maintains and enhances brain function through precisely timed electrical rhythms that coordinate the neural activities essential for memory, restoration, and cognitive health. Every night as you surrender to sleep, your brain embarks on one of its most crucial tasks, transforming the fleeting experiences of the day into lasting memories that will shape who you are tomorrow. This process, called memory consolidation, represents one of sleep's most vital functions and helps explain why a good night's rest can mean the difference between forgetting a lesson and mastering it completely. The star performers in this nightly memory theater are the hippocampus and the cerebral cortex, two brain regions that engage in an intricate dialogue throughout the hours of sleep. The hippocampus, a seahorse-shaped structure buried deep within the temporal lobe, serves as your brain's temporary storage facility. During wakefulness, it rapidly encodes new experiences, creating fragile memory traces that require further processing to become permanent. The cortex, meanwhile, houses your brain's vast library of established knowledge and long-term memories. During sleep, these two regions engage in a sophisticated conversation that transfers important information from temporary hippocampal storage to more permanent cortical locations. This transfer process is not random but highly selective, prioritizing memories that are emotionally significant, repeatedly encountered or tagged as important for future use. Slow-wave sleep, the deep NREM stage, characterized by powerful delta rhythms, serves as the primary vehicle for declarative memory consolidation. Declarative memories encompass facts, events, and conscious recollections that you can verbally describe. The name of your first-grade teacher, the plot of yesterday's movie, the capitals of European countries. These are all declarative memories that rely on slow-wave sleep for their permanent storage. During deep sleep, the hippocampus systematically replays the day's experiences, firing in the same sequences that occurred during learning. These replay events happen at an accelerated pace, compressing hours of experience into brief bursts of neural activity. As the hippocampus replays these memory traces, the cortex listens and responds, gradually incorporating the new information into its existing knowledge networks. Research using sophisticated brain imaging techniques has revealed that this hippocampal cortical dialogue occurs in precisely timed waves. Sharp wave ripples, brief bursts of high-frequency activity in the hippocampus, coincide with sleep spindles and slow oscillations in the cortex. This temporal coordination ensures that the cortex is in the optimal state to receive and integrate new information from the hippocampus. The strength of this consolidation process directly correlates with sleep quality and duration. People who obtain adequate slow-wave sleep show significantly better retention of factual information learned before bedtime compared to those who are sleep-deprived or whose deep sleep is disrupted. Even a single night of poor sleep can impair memory consolidation, while chronic sleep deprivation can severely compromise the ability to form lasting memories. REM sleep, with its vivid dreams and heightened brain activity, serves a different but equally important role in memory processing. While slow-wave sleep handles factual information, REM sleep specializes in consolidating procedural memories, the unconscious skills and habits that guide your daily actions. Learning to ride a bicycle, mastering a musical instrument, or developing the muscle memory for typing all depend on REM sleep for their consolidation. During REM sleep, brain regions involved in motor control, skill learning, and emotional processing become highly active. The cerebellum, which coordinates movement and balance, shows increased communication with motor cortex areas, strengthening the neural pathways that underlie skilled movements. This is why athletes and musicians often find that complex skills improve after a good night's sleep, even without additional practice, REM sleep also plays a crucial role in creativity and insight formation. The unique neurochemical environment of REM sleep, characterized by high acetylcholine levels and reduced norepinephrine, allows for novel connections between disparate pieces of information. Many scientists and artists have reported breakthrough insights occurring after sleep, and research confirms that REM sleep enhances creative problem-solving and the ability to see unexpected relationships between concepts. The replay of daily experiences during sleep extends beyond simple repetition. Neural circuits engaged during learning show spontaneous reactivation during both NREM and REM sleep, but this reactivation is not merely a passive echo of waking activity. Instead, the sleeping brain actively processes and transforms these experiences, extracting general principles, identifying patterns, and integrating new information with existing knowledge. Studies sophisticated recording techniques have shown that place cells in the hippocampus, which fire when an animal is in specific locations, replay entire spatial sequences during sleep. A rat that spent the day navigating a maze will show the same sequence of place cell activation during sleep, as if mentally retracing its steps. This replay occurs at accelerated speeds and in both forward and reverse directions, suggesting that the brain is not simply reviewing experiences but actively analyzing and organizing them. The molecular mechanisms underlying memory consolidation during sleep involve widespread changes in synaptic strength throughout the brain. Sleep promotes the synthesis of proteins necessary for long-term memory formation, while also facilitating the selective weakening of less important synaptic connections. This process, known as synaptic homeostasis, prevents the brain from becoming oversaturated with information while strengthening the most relevant memories. During slow-wave sleep, the brain exhibits reduced overall activity combined with synchronized oscillations that promote synaptic plasticity. These conditions favor the consolidation of important memories while allowing weaker memory traces to fade. The regulation of synaptic strength during sleep helps maintain the brain's capacity to learn new information while preserving the most valuable memories from previous experiences. Sleep's role in forgetting is as important as its role in remembering. The brain cannot store every detail of daily experience without becoming overwhelmed with irrelevant information. Sleep facilitates active forgetting, a process that selectively eliminates unnecessary memories while preserving important ones. This memory pruning occurs through the systematic weakening of synaptic connections that encode trivial or redundant information. Research has shown that sleep deprivation impairs both remembering and forgetting, suggesting that these processes are interconnected aspects of memory maintenance. People who are chronically sleep deprived often report feeling mentally cluttered, unable to distinguish important information from irrelevant details. This cognitive fog may result from impaired memory pruning processes that normally occur during healthy sleep. The connection between sleep quality and learning performance has profound implications for education and cognitive development. Students who maintain regular sleep schedules and obtain adequate deep sleep show superior academic performance compared to those with irregular or insufficient sleep. The timing of sleep relative to learning also matters, with sleep occurring within hours of learning providing the greatest memory benefits. Napping can also enhance memory consolidation, particularly when the nap includes slow-wave sleep. Even brief periods of deep sleep can improve retention of recently learned information, though longer naps that include both NREM and REM sleep provide more comprehensive memory benefits. The optimal nap duration appears to be 60 to 90 minutes, allowing for one complete sleep cycle. Understanding the relationship between sleep and memory consolidation reveals why sleep should be considered an essential component of the learning process, rather than time taken away from study. The sleeping mind continues the work of learning, transforming daily experiences into lasting knowledge and skills through the remarkable process of memory consolidation that occurs every night. Your brain generates waste products constantly throughout the day. Every thought, every memory, every moment of consciousness creates cellular debris that must be cleared away to maintain optimal function. For decades, scientists puzzled over how the brain accomplished this essential housekeeping task. Unlike other organs that rely on lymphatic vessels to drain waste, the brain appeared to lack such a system entirely. This mystery persisted until 2012, when researchers at the University of Rochester made a groundbreaking discovery that revolutionized our understanding of sleep's biological purpose. Using advanced imaging techniques, neuroscientist Maiken Nedergaard and her team revealed that the brain possesses its own specialized waste clearance system, which they named the glymphatic system. This network relies on cerebrospinal fluid, the clear liquid that bathes the brain and spinal cord, to flush away metabolic waste products. The system earned its name from the crucial role played by glial cells, particularly astrocytes, in facilitating this cleaning process. The glymphatic system operates like a sophisticated plumbing network, but one that becomes dramatically more active during sleep. During wakefulness, this waste clearance system functions at reduced capacity, processing only a fraction of the brain's accumulated cellular debris. However, when you fall asleep, particularly during deep non-REM sleep, the glymphatic system springs into action with remarkable efficiency. The key to this sleep-dependent activation lies in the behavior of astrocytes, star-shaped brain cells that were once considered mere support structures, but are now recognized as active participants in brain function. During wakefulness, astrocytes maintain their normal size and shape, leaving relatively narrow channels between brain cells for cerebrospinal fluid to flow through. When sleep begins, these astrocytes undergo a dramatic transformation, shrinking by up to 60% of their normal volume. This astrocytic shrinkage creates wider pathways between neurons, effectively opening the brain's highways for waste removal. Cerebrospinal fluid can now surge through these expanded channels with much greater force and volume, carrying away accumulated toxins and metabolic byproducts. The result is a dramatic increase in waste clearance efficiency, with the sleeping brain removing waste at rates up to twice those seen during wakefulness. The cerebrospinal fluid that drives this cleaning process follows specific anatomical pathways through the brain. It enters along arteries, flows through the expanded extracellular spaces between neurons, and exits along veins, carrying dissolved waste products toward the lymphatic vessels in the neck. This flow is driven partly by arterial pulsations from the heartbeat and partly by the rhythmic breathing patterns that create pressure changes in the cerebrospinal fluid. Among the most important waste products cleared by the glymphatic system are amyloid beta and tau proteins, two substances strongly linked to Alzheimer's disease and other neurodegenerative conditions. Amyloid beta is produced naturally by neurons during normal activity, but when it accumulates faster than it can be cleared, it forms the characteristic plaques found in Alzheimer's brains. Similarly, tau protein, which normally helps maintain neuronal structure, can become misfolded and form tangles that disrupt cellular function. Research has shown that the glymphatic system clears amyloid beta at dramatically higher rates during sleep compared to wakefulness. In laboratory studies, sleeping mice showed amyloid beta clearance rates that were 60% higher than those observed in awake animals. This finding provides a compelling biological explanation for why chronic sleep deprivation increases the risk of developing Alzheimer's disease and other forms of dementia. The clearance of tau protein follows similar patterns, with sleep-dependent removal helping to prevent the accumulation of toxic tau aggregates. Studies in humans using advanced brain imaging techniques have confirmed that people with chronic insomnia show elevated levels of both amyloid beta and tau proteins in brain regions critical for memory and cognition. Beyond removing toxic proteins, sleep also facilitates the clearance of numerous other waste products generated by normal brain metabolism. These include lactate, a byproduct of glucose metabolism, and various inflammatory molecules that can impair neural function if allowed to accumulate. The efficient removal of these substances during sleep helps maintain the optimal chemical environment necessary for healthy brain function. The role of astrocytes in this cleaning process extends far beyond simply shrinking to create wider fluid channels. These versatile cells actively participate in waste processing through specialized water channels called aquaporin 4 channels. These molecular gates regulate the flow of cerebrospinal fluid through brain tissue and become more active during sleep, further enhancing waste clearance efficiency. Astrocytes also coordinate with other brain cells to optimize the timing and effectiveness of waste removal. They respond to sleep-related neurotransmitters like norepinephrine, which decreases during sleep, and signals astrocytes to begin their shrinkage process. This neurochemical coordination ensures that waste clearance is synchronized with other sleep-dependent processes. Sleep also triggers extensive cellular repair and protein synthesis throughout the brain. During deep sleep, neurons ramp up production of essential proteins needed for synaptic function, cellular structure, and metabolic processes. Many of these proteins cannot be synthesized efficiently during wakefulness due to the high energy demands of conscious brain activity. The protein synthesis that occurs during sleep is highly selective, prioritizing molecules essential for memory consolidation, synaptic plasticity, and cellular maintenance. Ribosomes, the cellular machinery responsible for protein production, show increased activity during sleep, particularly in brain regions that were highly active during the preceding day. This targeted protein synthesis helps repair damage caused by daily neural activity and prepares neurons for optimal function upon awakening. Mitochondrial maintenance represents another crucial aspect of sleep's cellular housekeeping functions. These cellular powerhouses, which generate the energy needed for all brain activity, accumulate damage throughout the day from the reactive oxygen species produced during energy metabolism. Sleep provides an opportunity for mitochondrial repair and renewal through processes that cannot occur efficiently during wakefulness. During sleep, mitochondria undergo fission and fusion processes that help maintain their health and efficiency. Damaged mitochondrial components are removed through selective autophagy, while new mitochondrial proteins are synthesized to replace worn-out structures. This maintenance is particularly important in the brain, which consumes about 20% of the body's total energy despite representing only 2% of body weight. The energy restoration that occurs during sleep also involves the replenishment of glycogen stores in the brain. Glycogen, the brain's stored form of glucose, becomes depleted during wakefulness and must be restored during sleep to maintain optimal cognitive function. Astrocytes play a key role in this process, storing glycogen and releasing glucose to neurons as needed. The connection between sleep quality and neurodegenerative disease risk has become increasingly clear through population studies and clinical research. People who consistently obtain less than 6 hours of sleep per night show accelerated cognitive decline and higher rates of dementia compared to those who sleep 7 to 8 hours. Similarly, individuals with sleep disorders like sleep apnea demonstrate increased accusation of Alzheimer's-related proteins in their brains. These findings suggest that chronic sleep deprivation may accelerate brain aging by impairing the glymphatic system's waste clearance functions. When toxic proteins accumulate faster than they can be removed, they begin to interfere with normal neural function, potentially triggering the cascade of events that leads to neurodegeneration. Understanding the brain's sleep-dependent cleaning systems has profound implications for preventing cognitive decline and maintaining brain health throughout life. The glymphatic system represents one of sleep's most essential functions, transforming those hours of apparent inactivity into a period of intensive neural maintenance that helps preserve cognitive function and protect against disease. Sleep is not a solitary performance by the brain alone, but rather a carefully choreographed symphony involving an entire orchestra of hormones that ebb and flow throughout the night. These chemical messengers coordinate the complex biological processes that make sleep both possible and restorative, orchestrating everything from the initial onset of drowsiness to the metabolic changes that occur during different sleep stages. The conductor of this hormonal symphony is melatonin, often called the sleep hormone, though its role extends far beyond simply making you feel tired. Produced by the pineal gland, a tiny pine cone-shaped structure buried deep within the brain, melatonin serves as your body's primary timekeeper, synchronizing internal biological rhythms with the external cycle of day and night. Melatonin synthesis follows a precise 24-hour schedule, controlled by the suprachiasmatic nucleus, your brain's master clock. As evening approaches and darkness falls, specialized light-sensitive cells in your retina detect the absence of blue light and send signals to the pineal gland to begin melatonin production. Blood levels of this hormone rise dramatically, typically starting around 9 in the evening, reaching peak concentrations between 2 and 4 in the morning, then falling rapidly as dawn approaches. This melatonin surge does more than simply induce sleepiness. It triggers a cascade of physiological changes that prepare your body for sleep. Core body temperature begins to drop, blood pressure decreases, and alertness wanes. Melatonin also influences other hormone systems, setting the stage for the complex biochemical events that will unfold throughout the night. Modern life, with its abundance of artificial light, especially blue light from electronic devices, can suppress melatonin production and disrupt this natural rhythm, explaining why screen time before bed can interfere with sleep quality. Growth hormone represents one of sleep's most dramatic hormonal events, with the majority of daily growth hormone secretion occurring during deep NEREM sleep. The pituitary gland releases massive pulses of this hormone during the first few hours of sleep, when slow-wave sleep is most prevalent. In children and adolescents, this sleep-dependent growth hormone release is essential for physical development, bone growth, and tissue maturation, Adults continue to experience sleep-related growth hormone surges throughout life, though the amounts decrease with age. This hormone promotes protein synthesis, tissue repair, and cellular regeneration across the body. It facilitates muscle growth and repair, aids in wound healing, and supports immune system function. The growth hormone released during sleep also affects metabolism, promoting the breakdown of fat stores and helping to maintain healthy body composition. The timing of growth hormone release is tightly linked to sleep architecture, with the largest pulses occurring during the deepest stages of NREM sleep. Sleep deprivation dramatically reduces growth hormone secretion, which may explain why chronically sleep-deprived individuals often struggle with tissue repair, immune function, and metabolic regulation. This connection highlights why adequate sleep is particularly crucial during periods of growth, healing, or physical stress. Cortisol, commonly known as the stress hormone, follows a pattern that is essentially opposite to melatonin. This hormone, produced by the adrenal glands, typically reaches its lowest levels during the early hours of sleep and begins rising in the pre-dawn hours, reaching peak concentrations shortly after awakening. This cortisol awakening response helps mobilize energy stores and prepare the body for the metabolic demands of the coming day. During healthy sleep, cortisol levels remain suppressed, allowing for the restorative processes that occur during rest. However, chronic stress, sleep disorders, or irregular sleep schedules can disrupt normal cortisol patterns, leading to elevated nighttime cortisol levels that interfere with sleep initiation and maintenance. High cortisol during sleep can fragment sleep architecture, reduce time spent in deep NREM stages, and impair memory consolidation processes. The relationship between sleep and cortisol creates a potentially vicious cycle. Poor sleep elevates stress hormone HELMO, while elevated stress hormones make it more difficult to achieve quality sleep. This interaction helps explain why stress management and good sleep hygiene are essential for maintaining healthy cortisol rhythms and optimal sleep quality. Sleep profoundly affects insulin sensitivity and glucose metabolism, with significant implications for metabolic health. During sleep, particularly during deep NREM stages, insulin sensitivity increases, allowing cells to more efficiently process glucose and maintain stable blood sugar levels. This enhanced insulin sensitivity during sleep helps optimize energy storage and utilization throughout the body. Sleep deprivation disrupts these metabolic processes in multiple ways. Even a single night of poor sleep can reduce insulin sensitivity by up to 25%, making it more difficult for cells to respond to insulin, and potentially leading to elevated blood glucose levels. Chronic sleep deprivation increases the risk of developing type 2 diabetes and metabolic syndrome, highlighting the crucial connection between sleep quality and metabolic health. The hormones leptin and ghrelin work in tandem to regulate appetite and energy balance, and both are significantly affected by sleep patterns. Leptin, produced by fat cells, signals satiety to the brain and helps suppress appetite when energy stores are adequate. Ghrelin, produced primarily in the stomach, stimulates hunger and promotes food intake when energy is needed. Sleep restriction dramatically alters the balance between these appetite-regulating hormones. Poor sleep reduces leptin levels while increasing ghrelin production, creating a hormonal environment that promotes overeating and weight gain. People who consistently sleep less than 6 hours per night show leptin levels that are 18% lower and ghrelin levels that are 28% higher than those who sleep 8 hours nightly. This hormonal disruption helps explain why sleep deprivation is strongly associated with obesity and weight gain. The altered appetite signaling caused by inadequate sleep can lead to increased caloric intake, particularly cravings for high carbohydrate and high fat foods. The metabolic consequences of sleep loss extend beyond appetite regulation to affect fat storage, muscle maintenance, and overall energy expenditure. Temperature regulation during sleep involves complex hormonal coordination that affects both sleep quality and metabolic function. Core body temperature naturally decreases during sleep onset, reaching its lowest point during deep NREM sleep. This temperature drop is facilitated by hormonal changes, including increased production of vasodilating substances that promote heat loss through the skin. The sleep-related decrease in body temperature serves multiple functions. It reduces metabolic rate, conserving energy for restorative processes. It also appears to facilitate the deeper stages of NREM sleep, and may contribute to memory consolidation processes. Disruptions in normal temperature regulation, whether from environmental factors, illness, or hormonal imbalances, can significantly impair sleep quality and architecture. Hormonal disruptions can profoundly affect sleep quality, through various mechanisms. Thyroid disorders alter metabolic rate and temperature regulation, affecting sleep initiation and maintenance. Reproductive hormone fluctuations during menstrual cycles, pregnancy and menopause can disrupt sleep patterns through their effects on temperature regulation and neurotransmitter systems. Age-related changes in hormone production contribute to the sleep difficulties commonly experienced by older adults. Understanding sleep's hormonal orchestra reveals why maintaining healthy sleep patterns is essential for overall physiological function. These hormonal systems work together to coordinate the complex biological processes that occur during sleep, from growth and repair to metabolic regulation and appetite control. Disruption of any component of this system can have cascading effects on sleep quality and overall health. REM sleep represents one of the most paradoxical states in all of neuroscience. Your body lies motionless, yet your brain races with activity that rivals the most intense moments of wakefulness. Your muscles are paralyzed, yet your mind soars through vivid landscapes of impossible physics and forgotten memories. This strange contradiction between physical stillness and mental turbulence has fascinated researchers for decades, revealing a sleep state so unique that it almost seems like a separate form of consciousness entirely. The neurochemistry of REM sleep creates an internal environment unlike any other experienced by the human brain. During this phase, acetylcholine, a neurotransmitter crucial for attention and learning, surges to levels that match or exceed those seen during peak wakefulness. This acetylcholine flood originates from clusters of neurons in the brainstem, particularly in regions called the lateral or dorsal tegmental nucleus and the pedunculopontine tegmental nucleus, which act as REM sleep's primary control centers. Simultaneously, other key neurotransmitters virtually disappear from the brain. Norepinephrine, dopamine, and serotonin, chemicals that normally help maintain logical thinking and emotional regulation during wakefulness, drop to their lowest levels of the entire 24-hour cycle. This dramatic neurochemical shift creates a brain state characterized by heightened acetylcholine activity combined with the near absence of modulatory neurotransmitters, producing the distinctive mental landscape of dreams. The high acetylcholine levels during REM sleep activate brain regions involved in learning, memory, and sensory processing, while simultaneously disrupting the normal communication between the prefrontal cortex and other brain areas. The prefrontal cortex, responsible for logical reasoning, self-awareness, and critical thinking, becomes effectively disconnected from the rest of the brain. This disconnection explains why dreams often feature bizarre storylines, impossible events, and a striking lack of self-critical awareness that would normally question such absurdities. The paralysis that accompanies REM sleep serves as a crucial protective mechanism, preventing you from physically acting out the complex movements and behaviors occurring in your dreams. This rhaminotonia, as neuroscientists call it, results from active inhibition of motor neurons by brainstem circuits that release the neurotransmitter GABA. These inhibitory signals essentially disconnect the brain's motor commands from the spinal cord, creating a temporary paralysis that affects all voluntary muscles except the diaphragm, which continues breathing, and the extraocular muscles that control eye movements. The brainstem regions responsible for rhaminotonia include the sublateral dorsal nucleus and the ventromedial medulla, which send inhibitory signals down the spinal cord to suppress motor neuron activity. This system is so effective that even the most vigorous dream activities, from running through forests to fighting monsters, produce no actual movement beyond the characteristic rapid eye movements that give this sleep stage its name. Occasionally, this paralysis system can malfunction, leading to REM sleep behavior disorder, a condition where people physically act out their dreams. This disorder provides compelling evidence for the normal protective function of rhaminotonia and offers insights into the neural mechanisms that coordinate the relationship between dream content and motor control. Dreams during REM sleep appear to serve important functions in emotional processing and psychological well-being. The unique neurochemical environment of REM sleep, with its reduced norepinephrine and heightened acetylcholine, creates optimal conditions for processing emotional memories while reducing their associated stress response. The amygdala, the brain's emotion center, remains highly active during REM sleep, allowing for the reprocessing of emotional experiences without the high levels of stress hormones that accompanied the original events. Research suggests that REM sleep helps integrate emotional experiences with existing memories, potentially reducing the emotional intensity of traumatic or stressful events. People who are deprived of REM sleep often show increased emotional reactivity and difficulty processing negative experiences. This emotional regulation function of dreaming may explain why individuals with depression and anxiety disorders frequently experience disrupted REM sleep patterns. The bizarre and often illogical nature of dreams may actually facilitate creative problem-solving and insight formation. The reduced activity in brain regions responsible for logical constraints allows for novel connections between disparate concepts and memories. Many scientific discoveries and artistic creations have reportedly emerged from dream experiences, suggesting that the unconstrained neural activity of REM sleep can generate insights unavailable to the waking mind. REM sleep shows dramatic developmental changes across the human lifespan, reflecting its important role in brain maturation and maintenance. Newborn infants spend approximately 50% of their sleep time in REM, compared to only 20 to 25% in healthy adults. This high proportion of REM sleep during early development suggests crucial roles in brain growth, neural pathway formation, and the establishment of proper neural connectivity. During fetal development, REM sleep appears to provide essential stimulation for the developing nervous system. The rapid eye movements and associated brain activity may help establish proper connections between neurons and promote the growth of neural pathways essential for sensory processing and motor control. Premature infants show even higher percentages of REM sleep, supporting the hypothesis that this sleep state plays a vital role in neural development. As people age, REM sleep typically decreases in both duration and intensity. Elderly individuals often show fragmented REM periods and reduced overall REM sleep time, changes that may contribute to age-related cognitive decline and emotional regulation difficulties. The age-related reduction in REM sleep coincides with decreased production of the neurotransmitters necessary for generating robust REM states. Lucid dreaming represents a fascinating variation of REM consciousness where the dreamer becomes aware that they are dreaming while remaining in the dream state. This phenomenon occurs in approximately 23% of people at least once per month, with some individuals developing the ability to achieve lucid dreams reliably. During lucid dreaming, parts of the prefrontal cortex, associated with self-awareness and metacognition, show increased activity compared to normal REM sleep, while the dreamer maintains the vivid sensory experiences characteristic of dreams. Lucid dreamers report being able to exert some degree of control over their dream content, from changing dream scenery to practicing skills or confronting fears in a safe environment. Research using EEG and brain imaging techniques has confirmed that lucid dreaming represents a hybrid state of consciousness, combining elements of both REM sleep and wakefulness in unique ways that expand our understanding of consciousness itself. Various techniques can increase the likelihood of experiencing lucid dreams, including reality checks during waking hours, maintaining dream journals, and specific cognitive strategies practiced before sleep. Some researchers are investigating whether lucid dreaming could be used therapeutically to treat nightmares or practice skills in a safe environment. Theories about dream function remain diverse and sometimes contradictory, reflecting the complexity of this unique brain state. The activation synthesis hypothesis proposes that dreams result from the brain's attempt to make sense of random neural activity generated during REM sleep. According to this theory, the bizarre content of dreams reflects the brain's efforts to create coherent narratives from essentially meaningless neural noise. The threat simulation theory suggests that dreams serve an evolutionary function by allowing us to practice responses to dangerous situations in a safe environment. Dreams often feature threatening scenarios, potentially helping to prepare neural and behavioral responses to real-world dangers. This theory helps explain why anxiety dreams and nightmares are common across cultures and throughout history. More recent theories emphasize dreams' role in memory consolidation and emotional regulation. The emotional regulation theory proposes that REM sleep allows for the reprocessing of emotional memories, reducing their emotional charge while preserving important information. The memory consolidation theory suggests that dreams reflect the brain's efforts to integrate new experiences with existing knowledge networks. The continual activation theory proposes that REM sleep serves a maintenance function, keeping neural circuits active and preventing them from degrading during periods of sensory deprivation. Dreams, according to this view, represent the subjective experience of this neural maintenance activity. Understanding REM sleep and dreaming reveals one of the most remarkable aspects of human consciousness: This nightly journey into a parallel universe of experience serves essential functions in memory processing, emotional regulation, and brain maintenance. The dreaming brain demonstrates the remarkable plasticity and creativity of neural systems, transforming the biochemical chaos of REM sleep into the rich tapestry of experiences we call dreams. Sleep disorders represent far more than mere inconveniences that interfere with rest. They serve as unique windows into the intricate mechanisms that govern normal sleep, revealing how specific disruptions in brain circuitry can unravel the carefully orchestrated processes that unfold each night. By studying what happens when sleep goes wrong, researchers have gained unprecedented insights into how the healthy brain achieves and maintains its various sleep states. Insomnia, the most common sleep disorder affecting up to 30% of adults, provides compelling evidence for the role of overactive brain networks in preventing sleep initiation and maintenance. Rather than simply representing an inability to fall asleep, chronic insomnia reflects a state of persistent hyperarousal, where the brain's wake-promoting systems remain inappropriately active during times when sleep should naturally occur. Neuroimaging studies of people with chronic insomnia reveal increased activity in brain regions associated with worry, rumination, and emotional processing. The anterior cingulate cortex and prefrontal cortex, areas normally suppressed during sleep onset, remain highly active in insomniacs, creating a mental state characterized by racing thoughts and heightened alertness. The amygdala, the brain's alarm center, also shows excessive activity, suggesting that chronic insomnia involves a persistent state of threat detection that prevents the relaxation necessary for sleep. The hyperarousal model of insomnia has revealed important insights into normal sleep mechanisms. Healthy sleep initiation requires the coordinated suppression of multiple wake-promoting brain networks, including those involved in attention, executive control, and emotional processing. When stress, anxiety, or other factors prevent this coordinated deactivation, the result is the frustrating experience of lying awake despite feeling physically tired. Sleep apnea affects millions of people worldwide and demonstrates how disrupted breathing during sleep can have profound effects on neural function. This disorder occurs when the upper airway repeatedly collapses during sleep, causing breathing interruptions that fragment sleep architecture and reduce oxygen delivery to the brain. Each apnea episode triggers a brief arousal that restores breathing but prevents the deeper stages of sleep necessary for optimal brain function. The repetitive hypoxia, or oxygen deprivation that characterizes sleep apnea, has cascading effects on neural health. Brain regions particularly sensitive to oxygen deprivation, including the hippocampus and prefrontal cortex, show structural changes in people with severe sleep apnea. These alterations correlate with the cognitive impairments commonly experienced by sleep apnea patients, including problems with memory, attention, and executive function. Sleep apnea also disrupts the glymphatic system's waste clearance functions, as the repeated arousals and altered sleep architecture interfere with the deep sleep stages when brain cleaning is most efficient. This disruption may contribute to the increased risk of dementia and other neurodegenerative diseases observed in people with untreated sleep apnea. Narcolepsy offers unique insights into REM sleep regulation by demonstrating what happens when the boundaries between sleep and wakefulness become blurred. This disorder results from the loss of neurons that produce hypocretin, also called orexin, a neuropeptide crucial for maintaining wakefulness and regulating sleep-wake transitions. Without adequate hypocretin signaling, the normal barriers between sleep states break down, allowing REM sleep phenomena to intrude into wakefulness. People with narcolepsy experience excessive daytime sleepiness and may suddenly enter REM sleep during the day, sometimes falling directly into dream states without passing through the normal NREM sleep stages. They may also experience cataplexy, a sudden loss of muscle tone triggered by strong emotions that resembles the muscle paralysis of REM sleep occurring during wakefulness. Sleep paralysis and hypnagogic hallucinations, other symptoms of narcolepsy, further demonstrate how REM sleep components can inappropriately activate during wake periods. Studying narcolepsy has revealed the critical role of hypocretin neurons in maintaining sleep-wake boundaries, and has led to new understanding of how different sleep states are normally kept separate and distinct. The discovery that narcolepsy often involves autoimmune destruction of hypocretin neurons has also provided insights into how the immune system can affect sleep regulation. Sleepwalking and other partial arousal disorders demonstrate that consciousness exists on a spectrum rather than as a simple binary state of sleep or wakefulness. These disorders, which include sleep talking, confusional arousals, and sleep terrors, occur when parts of the brain wake up while other regions remain in deep sleep. The motor cortex and subcortical movement centers become active enough to generate complex behaviors, while areas responsible for memory formation and conscious awareness remain in slow-wave sleep. Brain imaging during sleepwalking episodes reveals this mixed state of consciousness, with some brain regions showing the slow-wave activity characteristic of deep sleep, while others display patterns similar to wakefulness. This neurological evidence confirms that sleepwalking represents a genuine hybrid state rather than simply acting out dreams or conscious deception. The study of partial arousal disorders has enhanced understanding of how different brain regions can independently transition between sleep and wake states. This research suggests that consciousness itself may be more distributed and variable than traditionally thought, with different brain networks capable of operating in different states simultaneously. REM sleep behavior disorder presents a particularly informative window into the neural mechanisms of REM sleep and its relationship to neurodegenerative diseases. In this condition, the brainstem circuits that normally paralyze voluntary muscles during REM sleep fail to function properly, allowing people to physically act out their dreams. This disorder often involves violent or aggressive dream content, leading to injury of the affected person or their sleep partner. REM sleep behavior disorder is strongly associated with synucleinopathies, a group of neurodegenerative diseases that include Parkinson's disease, Lewy body dementia, and multiple system atrophy. The disorder often appears years or even decades before other symptoms of neurodegeneration, making it a potential early marker for these conditions. The brainstem regions affected in REM sleep behavior disorder overlap with areas where pathological protein aggregates first appear in these diseases. This connection has revealed important insights into how neurodegeneration progresses through the brain and has highlighted the vulnerability of sleep-regulating circuits to pathological processes. The study of REM sleep behavior disorder has also improved understanding of the normal mechanisms that maintain REM atonia and prevent dreaming. Approaches for sleep disorders target specific neural mechanisms revealed through research into these conditions. Continuous positive airway pressure therapy for sleep apnea works by preventing airway collapse and maintaining normal oxygenation, protecting the brain from hypoxic damage. Cognitive behavioral therapy for insomnia addresses the overactive brain networks that prevent sleep by teaching techniques to reduce cognitive and physiological arousal. Medications for various sleep disorders target specific neurotransmitter systems identified through sleep disorder research. Sodium oxybate for narcolepsy enhances deep sleep and consolidates nighttime sleep by affecting GABA signaling. Clonazepam for REM sleep behavior disorder suppresses REM sleep muscle activity by enhancing inhibitory neurotransmission. Understanding sleep disorders has revealed that healthy sleep depends on the coordinated function of multiple brain systems working in harmony. When any component of this complex system malfunctions, the resulting disorders provide valuable insights into the normal mechanisms that allow us to transition smoothly between wakefulness and sleep, maintain appropriate sleep architecture, and awaken refreshed and cognitively restored. Sleep represents one of evolution's greatest paradoxes. In a world where constant vigilance often means the difference between life and death, why would natural selection favor a state that renders animals unconscious, immobile, and vulnerable to predators? For 1/3 of every day, creatures across the animal kingdom voluntarily enter a condition that seems to violate every principle of survival. They cannot flee from danger, cannot search for food, cannot defend their territory, and cannot reproduce. Yet despite these obvious disadvantages, sleep has persisted across millions of years of evolution, suggesting that its benefits must be so profound that they outweigh its considerable risks. The universality of sleep across the animal kingdom provides compelling evidence for its fundamental importance. Every animal with a complex nervous system sleeps in some form, from the smallest insects to the largest mammals. Fruit flies require their daily rest periods and show many of the same sleep characteristics as humans, including rebound sleep after deprivation and increased sleep following learning experiences. Even jellyfish, which lack a centralized brain, exhibit sleep-like states with reduced activity and responsiveness. This universality suggests that sleep evolved early in the history of complex life and has been so advantageous that no lineage has successfully abandoned it. The fact that sleep appears across such diverse species, from creatures that live in constant danger to those at the top of the food chain, indicates that the benefits of sleep transcend specific ecological niches or survival strategies. The relationship between brain size and sleep requirements reveals intriguing patterns that provide clues about sleep's evolutionary functions. Generally, animals with larger brains relative to their body size tend to sleep more than those with smaller brains. Humans, with their exceptionally large brains, require about 8 hours of sleep per day. Elephants, despite their massive brains in absolute terms, sleep only 4 to 6 hours daily, but their brain-to-body ratio is much smaller than that of humans. This pattern suggests that sleep serves functions specifically related to brain maintenance and cognitive processing rather than simple physical rest. The metabolic demands of large, complex brains may require extended periods of reduced activity to allow for cellular repair, waste clearance, and memory consolidation. Species with simpler nervous systems can maintain their neural functions with less intensive maintenance periods. Marine mammals have evolved perhaps the most remarkable solution to sleep's vulnerability problem through unihemispheric sleep, where only half the brain sleeps at a time while the other half remains alert. Dolphins, whales, and seals can literally sleep with one eye open, maintaining vigilance for predators while still obtaining necessary rest. During unihemispheric sleep, one brain hemisphere shows the slow wave activity characteristic of deep sleep, while the other remains in a wakeful state. This adaptation allows marine mammals to continue swimming to the surface to breathe, watch for predators, and maintain social contact with their pods, while still obtaining restorative sleep. The two brain hemispheres alternate their sleep periods, ensuring that each gets adequate rest over time. This remarkable adaptation demonstrates that evolution can find creative solutions to sleep's inherent vulnerabilities when the benefits are essential enough. The discovery of unihemispheric sleep also reveals important insights about sleep's core functions. If sleep served only simple restorative purposes, it might be possible for the entire brain to obtain these benefits simultaneously. The fact that marine mammals can achieve sleep's benefits with only half the brain at a time suggests that sleep involves local cellular processes that can occur independently in different brain regions. Birds present another fascinating case study in sleep evolution, showing remarkable diversity in sleep strategies. Many birds can sleep while perched on branches using a tendon-locking mechanism that prevents them from falling. Some species like frigate birds can sleep while flying, using unihemispheric sleep to maintain navigation while resting portions of their brain. Migratory birds can dramatically reduce their sleep during long flights, sometimes sleeping only a few minutes at a time for days or weeks. The ability of some bird species to function with minimal sleep during migration challenges our understanding of sleep's necessity. These birds appear to enter a state where brief microsleeps provide essential neural maintenance while allowing continued flight. Upon reaching their destinations, however, these birds typically show intense rebound sleep, suggesting that even they cannot indefinitely postpone sleep's restorative functions. Reptiles and amphibians show simpler sleep patterns than mammals and birds, but still demonstrate clear rest periods, with reduced responsiveness and specific sleeping postures. Even fish exhibit rest states, though whether these constitute true sleep comparable to that of higher vertebrates remains debated. The presence of rest states across such diverse vertebrate groups suggests that the fundamental need for periodic neural downtime appeared early in vertebrate evolution. The evolutionary costs of sleep are substantial and obvious. Sleeping animals cannot gather food, find mates, care for offspring, or defend against threats. Many species have evolved adaptations to minimize these costs, such as sleeping in protected locations, sleeping in groups where some individuals can maintain watch, or timing sleep periods to coincide with times when activity would be less productive. Despite these costs, the benefits of sleep must be even greater. Current evidence suggests these benefits include memory consolidation, cellular repair, waste clearance from the brain, immune system enhancement, and metabolic regulation. The fact that no animal has successfully evolved to eliminate sleep entirely suggests that these functions cannot be adequately performed during wakefulness. The persistence of sleep across evolutionary time and biological diversity points to fundamental constraints in how complex nervous systems operate. Sleep may represent an unavoidable consequence of having a brain sophisticated enough to process complex information, form memories, and maintain consciousness. The evolutionary mystery of sleep ultimately reveals that the benefits of consciousness and intelligence come with the price of vulnerability, a trade-off that countless species have found worthwhile across millions of years of natural selection. Sleep is not a static phenomenon that emerges fully formed at birth, but rather a dynamic process that evolves dramatically throughout the human lifespan. From the earliest moments of fetal development to the final years of life, sleep patterns undergo profound transformations that mirror the changing needs of the developing and aging brain. Understanding these changes reveals sleep's intimate relationship with neural maturation, and provides insights into why sleep requirements shift so dramatically as we progress through different stages of life. Sleep begins remarkably early in human development, appearing in the fetal brain around the 23rd week of gestation. At this stage, the developing nervous system starts exhibiting alternating periods of activity and quiescence that resemble the basic rhythms of sleep and wakefulness. Fetal sleep serves crucial functions in brain development, providing the neural stimulation necessary for proper formation of synaptic connections and the establishment of fundamental brain circuits. During fetal development, REM sleep dominates, comprising up to 80% of total sleep time. This abundance of REM sleep appears essential for the rapid brain growth occurring during the final months of pregnancy. The intense neural activity characteristic of REM sleep provides the stimulation needed for neurons to form appropriate connections, establish proper pathways, and develop the complex networks that will support learning, memory, and consciousness after birth. The high proportion of REM sleep during fetal development also supports the formation of sensory systems and motor pathways. The rapid eye movements, muscle twitches, and neural firing patterns of fetal REM sleep help establish the connections between sensory organs and brain regions, preparing these systems for the sensory-rich environment the infant will encounter after birth. Newborn infants continue to exhibit sleep patterns dramatically different from those of adults, spending 16 to 18 hours per day asleep, with REM sleep still comprising approximately 50% of total sleep time. This extended sleep duration reflects the enormous energy demands of rapid brain growth and the need for intensive neural development during the first months of life. Infant sleep architecture differs fundamentally from adult patterns in several key ways. Newborns often enter REM sleep directly from wakefulness, bypassing the progressive NREM stages that characterize mature sleep. Their sleep cycles are shorter, lasting about 50 to 60 minutes compared to the 90-minute cycles of adults. Sleep is also more fragmented, with frequent awakenings that may have evolutionary advantages by ensuring regular feeding and promoting parent-infant bonding. The gradual maturation of infant sleep patterns reflects underlying brain development. The emergence of clear NREM sleep stages corresponds to the maturation of thalamic circuits that generate sleep spindles and slow oscillations. The consolidation of nighttime sleep and the development of circadian rhythms parallel the maturation of the suprachiasmatic nucleus and its connections to other brain regions. By 6 months of age, most infants have developed more adult-like sleep patterns, with longer periods of consolidated nighttime sleep and more distinct sleep stages. However, REM sleep still comprises about 30% of total sleep time, reflecting the continued importance of this sleep stage for ongoing neural development and learning. Adolescence brings another dramatic transformation in sleep patterns, characterized by shifts in both sleep timing and architecture. The biological changes of puberty trigger alterations in circadian rhythm regulation, causing teenagers to naturally fall asleep later and wake up later than children or adults. This shift, known as delayed sleep phase, results from changes in melatonin secretion patterns and altered sensitivity to light exposure. The adolescent brain undergoes extensive remodeling during this period, with processes of synaptic pruning eliminating unnecessary neural connections while strengthening important pathways. Sleep plays a crucial role in this neural refinement, with slow-wave sleep facilitating the elimination of weak synaptic connections while preserving and strengthening those that are frequently used. Unfortunately, the biological tendency toward later sleep timing often conflicts with early school start times, creating a situation where many adolescents experience chronic sleep deprivation. This sleep loss occurs during a critical period of brain development and can impair academic performance, emotional regulation, and decision-making abilities. The recognition of these biological changes has led to calls for later school start times to better align with adolescent sleep needs. Adult sleep patterns typically stabilize in the 20s and remain relatively consistent through middle age. Healthy adults generally require 7 to 9 hours of sleep per night, with individual variations influenced by genetic factors, lifestyle, and health status. Adult sleep architecture shows the full complement of NREM and REM stages, with REM sleep comprising about 20 to 25% of total sleep time. During adulthood, sleep serves primarily maintenance functions rather than the developmental roles it played earlier in life. Memory consolidation, cellular repair, waste clearance, and hormonal regulation become the primary purposes of sleep, supporting cognitive performance and physical health throughout the adult years. As people enter their later decades, sleep undergoes another series of significant changes that reflect the aging process in the brain and body. Older adults typically experience earlier bedtimes and wake times, a shift that partly reverses the delayed phase preference of adolescence. Total sleep time often decreases, with many older adults sleeping only 6 to 7 hours per night. Age-related changes in sleep architecture are particularly striking. Deep REM sleep decreases substantially, sometimes virtually disappearing in very elderly individuals. Sleep becomes more fragmented, with frequent awakenings throughout the night. REM sleep may also decrease, though usually less dramatically than slow-wave sleep. These changes reflect alterations in the aging brain, including changes in neurotransmitter systems, reduced melatonin production, and alterations in circadian rhythm regulation. The suprachiasmatic nucleus shows decreased responsiveness to light cues, making it more difficult to maintain strong circadian rhythms. Age-related health conditions and medications can further disrupt sleep patterns. The evolution of sleep across the lifespan reveals its fundamental role in supporting the changing needs of the developing, mature, and aging brain. From the intensive neural development of early life through the maintenance functions of adulthood, to the challenges of aging, sleep patterns adapt to serve the brain's evolving requirements. Understanding these changes helps explain why sleep needs vary so dramatically at different life stages, and underscores the importance of age-appropriate sleep recommendations for optimal health and cognitive function throughout the human lifespan. Sleep deprivation exacts a toll on the human brain that is both immediate and cumulative, transforming the sharp, efficient organ that guides your daily life into a sluggish, error-prone version of itself. Even a single night of inadequate sleep triggers a cascade of neural changes that impair cognitive function, disrupt emotional regulation, and compromise decision-making abilities. The sleep-deprived brain becomes a shadow of its rested self, struggling to perform even basic tasks that would normally require minimal effort. The acute effects of sleep loss begin to manifest after just one night of reduced sleep, with measurable impairments appearing when sleep falls below 6 hours. The prefrontal cortex, the brain region responsible for executive functions like planning, reasoning, and impulse control, shows the most dramatic sensitivity to sleep loss. Brain imaging studies reveal that sleep-deprived individuals exhibit reduced activity in prefrontal regions, creating a neurological basis for the poor judgment and impaired decision-making that characterize sleep deprivation. Attention systems suffer particularly severe disruption during sleep deprivation. The ability to sustain focus on a task deteriorates rapidly as sleep debt accumulates, with attention becoming increasingly fragmented and prone to lapses. These attention deficits are not simply matters of reduced motivation or effort, but reflect fundamental changes in brain network connectivity that make sustained concentration neurologically difficult. Working memory, the mental workspace that holds and manipulates information for short periods, shows marked impairment even after moderate sleep restriction. Sleep-deprived individuals struggle to hold multiple pieces of information in mind simultaneously, and have difficulty manipulating that information to solve problems or make decisions. This working memory impairment affects everything from following complex instructions to performing mental arithmetic. Cognitive flexibility, the ability to adapt thinking patterns to changing circumstances, becomes severely compromised during sleep deprivation. The sleep-deprived brain tends to become stuck in rigid thinking patterns, struggling to generate creative solutions or adapt to unexpected situations. This inflexibility can have serious consequences in situations requiring rapid adaptation or innovative problem-solving. Decision-making abilities deteriorate in multiple ways during sleep deprivation. The brain begins to favor risky choices over conservative ones, seeking immediate rewards while ignoring long-term consequences. This shift toward risky decision-making reflects altered activity in brain regions that evaluate potential outcomes and weigh costs against benefits. Sleep-deprived individuals often make choices they would never consider when well-rested, from poor financial decisions to dangerous driving behaviors. The emotional consequences of sleep deprivation are equally profound and often more immediately noticeable than cognitive impairments. The amygdala, the brain's emotion center, becomes hyperreactive during sleep deprivation, responding more intensely to negative stimuli and showing reduced regulation by prefrontal control centers. This emotional dysregulation manifests as increased irritability, anxiety, and mood swings that constrain relationships and impair social functioning. Sleep loss disrupts the normal communication between emotional processing centers and areas responsible for emotional regulation. The result is a brain that experiences emotions more intensely, while simultaneously having reduced capacity to manage those emotions effectively. Minor frustrations become major irritations, small setbacks feel catastrophic, and positive emotions become more difficult to sustain. The relationship between sleep and mood is bidirectional and can create vicious cycles of sleep disruption and emotional distress. Poor sleep contributes to negative mood states, while anxiety and depression make it more difficult to achieve quality sleep. This interaction helps explain why sleep disorders are so commonly associated with mood disorders, and why treating sleep problems can often improve emotional well-being. Chronic sleep deprivation, defined as consistently obtaining less than the recommended 7 to 9 hours of sleep per night for adults, leads to long-term neural consequences that extend far beyond temporary impairments. Prolonged sleep restriction causes structural changes in the brain, including reduced gray matter volume in regions critical for cognitive function and emotional regulation. The hippocampus, essential for memory formation, shows particular vulnerability to chronic sleep loss. Studies have found that people with chronic insomnia exhibit reduced hippocampal volume, which correlates with memory deficits and may contribute to the increased risk of dementia associated with long-term sleep deprivation. These structural changes suggest that chronic sleep loss may accelerate brain aging and increase susceptibility to neurodegenerative diseases. Chronic sleep deprivation also disrupts the brain's waste clearance systems, leading to the accumulation of toxic proteins associated with Alzheimer's disease and other neurodegenerative conditions. The glymphatic system, which clears metabolic waste from the brain primarily during deep sleep, operates at reduced efficiency when sleep is chronically restricted, potentially contributing to the pathological protein accumulations that characterize these diseases. The brain attempts to compensate for sleep loss through various mechanisms, though these compensatory efforts are ultimately inadequate. During acute sleep deprivation, some individuals can temporarily maintain performance through increased effort and activation of additional brain regions not normally required for specific tasks. This neural compensation comes at a metabolic cost, however, and cannot be sustained indefinitely. Microsleeps represent another compensatory mechanism, where the sleep-deprived brain briefly enters sleep-like states lasting seconds to minutes while the person remains ostensibly awake. These involuntary sleep episodes allow for minimal neural restoration but create dangerous situations when they occur during activities requiring sustained attention, such as driving or operating machinery. The brain also attempts to prioritize the most essential sleep stages during recovery from sleep deprivation. When given the opportunity to sleep after deprivation, the brain shows rebound increases in slow-wave sleep, the deepest stage of NREM sleep most critical for restoration. This sleep rebound demonstrates the brain's recognition of sleep debt and its attempt to rapidly restore essential functions. Recovery sleep can reverse many of the acute effects of sleep deprivation, but complete restoration requires more than simply sleeping longer after a period of sleep loss. The brain needs multiple nights of adequate sleep to fully recover from extended sleep deprivation, and some effects of chronic sleep restriction may require weeks or months of improved sleep to resolve completely. Individual differences in sleep deprivation tolerance reflect genetic variations in sleep regulation and circadian rhythm control. Some people carry genetic variants that allow them to function adequately on less sleep than average, though these true short sleepers represent only about 1% of the population. Most people who believe they can function well on minimal sleep are actually experiencing chronic sleep deprivation without full awareness of their impaired performance. Age influences sleep deprivation tolerance, with older adults often experiencing more severe cognitive effects from sleep loss despite naturally requiring slightly less sleep. Adolescents and young adults may show greater resilience to acute sleep deprivation but remain vulnerable to the long-term consequences of chronic sleep restriction. Understanding the price of sleep deprivation reveals why sleep should be considered a non-negotiable biological necessity rather than an optional activity that can be sacrificed for other pursuits. The brain's vulnerability to sleep loss underscores the fundamental importance of prioritizing adequate sleep for cognitive function, emotional well-being, and long-term neurological health. After decades of research into the sleeping brain, we have uncovered remarkable insights into this nightly transformation that occurs in your skull. We now understand how memories consolidate through the intricate dialogue between hippocampus and cortex. We can trace the glymphatic system as it flushes toxic proteins from neural tissue. We have mapped the hormonal orchestras that coordinate repair and restoration. Yet sleep continues to guard many of its deepest secrets. We understand the mechanics of memory consolidation, but remain uncertain about consciousness itself during dreams. We can map the brain's waste clearance system but cannot fully explain why this essential cleaning cannot occur during wakefulness. Most intriguingly, we still cannot definitively answer why sleep evolved at all. In a world where vigilance means survival, why would natural selection favor a state of unconsciousness? The fact that every studied animal sleeps in some form suggests that sleep's benefits far outweigh its obvious risks. What remains clear is that sleep is not the opposite of wakefulness, but rather its essential partner. The brain that emerges each morning has been fundamentally altered by the night's journey through slow waves and rapid eye movements, cellular repair and memory processing, dreams and neurochemical cascades. Each sleep cycle writes and rewrites the neural connections that define who you are. As we continue to probe the mysteries of the sleeping brain, one truth becomes increasingly apparent. Those hours of apparent inactivity may represent some of the most important work our minds ever perform.